Hello, this topic about synapse, or in other way, how the signals from one neuron can be transmitted to the next neuron, and the major structure that link two neurons together called synapse. In the human body, there's not mainly, just two type of synapse: electric synapse and chemical synapse. In the case of electrical synapse, see there's a two neuron axon terminals, and these two neuron axon terminals are directly connected with each other by gap junction. So in these cases, ions. In the last topic we talked about ions. Say the major thing is sodium ions, the red dot sodium ions. And then sodium ions can this way and that way. In the case of the electron synapse, in many cases it's a function like the ions pass to mediate either this way or that way the propagation of the action potentials, just like in the same axon. So this is the electron signals is fast and bidirectional because the signal can go this way and that way. However, they are rare. It's not really that much existing in the nervous system. People believe there's a group of neurons in the center of the brain connect with each other. Not people believe, they observed. So they connect with electron neurons with each other, and then they believe they are really associated with, we call it the word, a synchronized activation of the same group of neurons, which are probably essential for our reasoning and other highly logical thinking, but it's on hypothesis. Another very typical synapse neurons, the electrical synapse, actually observed in the heart. If in the next semester you'll learn about the heart muscles and the electric synapse existing in each of the two cardiomyocytes. So, and this is about electron neurons. That's all we're going to talk about, the electron neurons. And in this topic, we'll focus on the chemical neurons. What is chemical neurons? The chemical neurons start with this is called presynaptic terminal ends. So this is basically the end of the axon terminals from a neuron that's existing upstream of this communication that we call presynaptic. And in the presynaptic neurons, you have this is granules, and all the granules, say, let me use some contain chemicals, and these chemicals have a name. They call it neurotransmitter. Later, neurotransmitter will be released outside the terminal, and the signal is transmitted from the presynaptic neurons to the next level, to the postsynaptic neurons. And this information is carried by neurotransmitters. And in addition, in the presynaptic neurons, you have also mitochondria, a lot of mitochondria. And because mitochondria, whatever, that's because this process itself need a lot of energy to happen. Each time of this release of the neurons need energy because, Is I mentioned? So this release of the pre-synaptic neurotransmitter is triggered by another thing, calcium. You will probably talk about calcium a lot. This is so what signals can trigger the neurotransmitters? Ultimately, action potential. When action potential propagate here, what the first thing that they do is that they release the calcium from internal storage of the calcium. And when calcium is released to the presynaptic terminal end, I'll use green one. Green, let me see. Bright blue, yes, I love this one. Right, green, 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 green. So this, the calcium trigger the, sorry, trigger the I call it granules to fuse into the surface of the memory and then release the grain. Where's my grain? Where's my grain here? And release the grain, not really grain, trying to say so. Release the neurotransmitter to the space. And this space actually has their own name. There is a very, say this one. This is called, this is the post synaptic. It can be a neuron or anything else, any other effector cells like muscle cells that can be activated by the synapse. They call it post-synaptic cells or whatever it is. And in between this is a very tiny, tiny, thin, narrow gap. This is called a cleft. It needs to be very thin and tiny because there's only a fixed number of neurotransmitters that can be released to the gap. And then the thinner the gap you have a higher concentration of the neurotransmitters. And then you have a higher potency to activate the postsynaptic effector cells or neurons. So, and this thin gap, we call it a cleft. And then on the postsynaptic neurons, you have let me use what color? Purple. It's purple. Okay. There has to be receptors. There has to be receptors on the surface of the neurons. So these receptors, if you remember, we just mentioned in the last chapter, there's two type of ion channels that can mediate the activation of the neurons by another neuron. One is ligand gated. See this. This is ligand gated. I'm sorry for my handwriting. My handwriting is not good, and it's even worse when I use this drawing pad. But anyway, this is ligand gated ion channels. If you remember, we talked about this in the last topic. This is fast, but not as strong. And in some cases, it's not, theoretically, ligand-gated ion channels does not say coexisting with the other, we call it, which color? I use a different color. Yeah, it's hard to find a color. Maybe this one. This is G protein coupled ion channels. And G protein once G protein is activated, go this way and that way, activate multiple channels. And G protein coupled ion channels is slower, but has higher potency. And normally, again, the ligand-gated ion channels and the G protein coupled ion channels does not coexisting in the same postsynaptic cells. it's easier for me to draw both of them in the same thing, but normally they're not. They're not existing in the same postsynaptic cells. And then that's it. That now you finish the transmission of signals from one neurons to the next neuron or the next effector cells by releasing the neurotransmitter. And what kind of signals is in most cases depend on what type of neurotransmitter you have right here. And then, so, okay, before we go, we're going to forward to talk about more about neurotransmitter. Before we doing that, there's another thing we have to mention. A very important also is it is important to always release the neurotransmitters. However, it's equally important to clear the neurotransmitters. The neurotransmitters clearance is extremely important because we not only want to promptly start our neural response, but we also want them to be stopped promptly. So, how many ways we can remove the neurotransmitters? First way, just by diffusion. In some cases, this cleft is open to the other tissue environment. So the neurotransmitters just go diffuse out from the cleft, and they gradually reduce the concentration. The interaction stops. And secondly, reuptake by synaptic neurons. So they can go back to the pre-synaptic terminal ends, and this is a fast way to remove it and also save something. I mean, save the materials you can use the next time. And the third way, so it may not be accurate, but to my best my understanding, it is a fast way. Which is this one, enzyme remove. And this enzyme, in most cases, is on the surface of the post-synaptic effector cells or neurons. It's in the post-synaptic. So here, I use different color, maybe red. So there's some enzyme here. They can remove, digest down the neurotransmitters. And one famous example is the skeletal muscles. Skeletal muscles have the enzymes to remove acetylcholine. Do I have acetylcholine in this topic? Probably no, because we are going to talk about acetylcholine in the future test. So I didn't introduce acetylcholine. So skeletal muscles are activated by acetylcholine, and skeletal muscles also have the enzymes to digest acetylcholine into acetyl and choline. So basically, that's a fast way you can remove the neurotransmitter as fast as possible, and then the cells, both the presynaptic, post-presynaptic, and the post-synaptic cells are ready for the next round activation. So that's about the structure of the chemical synapse and the electrical synapse. Next, let's talk about some major neurotransmitters. Let's say I don't really have a list of which one first, why this one's first or that one's later. But anyway, dopamine. Probably you have heard about dopamine. What's the function of the dopamine? Dopamine neurons that sensing dopamine is not really has a high abundance in our body, but the dopamine itself promote a very strong, very significant function in our body, in our behavior. Dopamine mediated the reward motivated behavior. And this the reward motivated behavior. What is this is extremely important for we learn things and we establish our behavior system. And so basically, there's people I find a reward center in the rat, but I don't think people find this in a human being. So basically, what is reward? Pleasure. Everything we do for the pleasure in our mind, and the reward itself is the pure pleasure. So basically, we are trying to do things that can activate the reward center to give us the pleasure, and that's motivate us to do things. And scientist has in my notes, I think I mentioned that scientist has made an experiment. Because they find the reward center in rats, they put an electrical probe inside the brain of the rat, and then the rat can control the pedal to keep activating, keep stimulating the reward center. Eventually, the rat die from thirsty and starving because once the rats find this mechanism, they keep pressing the pedal, no longer do anything else. It's just very addictive. So, and so again, dopamine mediate the reward motivated behavior. And for some, we don't know the details, but something we do, we can activate the dopamine in the brain, and this dopamine can activate some reward center in our brain. And also, dopamine can also help the muscle relaxation. So just imagine if you want to do any action like I'm doing it now, and writing and speaking and hold my hand, etc. Hold my head, etc. etc. So not only you need some muscle to contract, but also you need some muscle to relax. And those relaxation of muscles is mediated by the dopamine. And that's why if dopamine producing cells in the brain degenerated, when people do not produce enough dopamine, and a very famous example, the boxer, right? And the muscle become more stiff, so he cannot make the action more smoothly. So this is dopamine. Another thing, let me see, endorphin, and endorphin is the painkiller, painkiller, painkiller in our body. So dopamine, endorphin can endorphin can endorphin is released to say this synapse transmitting pain, and then endorphin is here. Endorphin gives the signal to the presynaptic neurons to block, to remove, to suppress. This is endorphin to suppress the pain transmission from this neuron to the next neuron. That's how the endorphin works. And then the receptor of the endorphin, we call it opioid receptor. You may heard the word of opioid. So this is the biggest issue because the opioid drugs, or I mean drugs, so they are basically doing the similar thing as in the activate the pathway endorphin, and now is the biggest issue like fentanyl killing millions of people every year, and dopamine, meth, I think meth, and cocaine is activated. The dopamine activate the pathway. I think meth and cocaine just block the clearance of dopamine, give people the pleasure, and both fentanyl, both now is a big issue. Of the United States. So this dopamine and endorphin. And let me see, what's the next one? Next one, let's talk about the. I think I can reuse this one. I'm gonna keep writing on this one. So this one is ade adenosine. Adenosine affect sleeping cycles. So let's say this daytime, this is nighttime. So during the daytimes, adenosine gradually increase. And then adenosine, its major function is to inhibit the activation of brain neurons. So during the daytime, the adenosine keep growing, and the neurons in the brain become less and less active. And at some moment, we just want to sleep. And this is a major control of a sleeping cycle, kind of. And then after sleeping, going down, the adenosine going down, and then wake up, going up, going down. It's sleep, and awake cycle, just like this. So this is basically how we feel sleepy. And then, like this, cocaine and meth and fentanyl can activate those way, but there's something can block this way, caffeine. Is it right? Caffeine, whatever. Can actually block this way. Can block adenosine. So adenosine is to inhibit the brain neurons. And then caffeine will bind to the adenosine receptor in the brain neurons, but it do nothing. It basically block adenosine there. And then the adenosine temporarily do not inhibit the brain, do not inhibit brain neuron activity temporarily. So in these cases, the people feel more excited, awake. However, caffeine does not really reduce the level of adenosine. So after the caffeine has been metabolized, the same adenosine are there, people still need to sleep. So basically caffeine, you're drinking coffee does not really let you to sleep less. You still need to sleep the same amount of time. However, you at that moment you can stay awake for longer. And interestingly, all of those are bad for sure, very bad. But caffeine, since it also just manipulates the neurotransmitters pathways, for many years people want to find out anything adverse effect of caffeine. But so far as I know, nothing has been discovered that any adverse effect from caffeine. And so far, the only very solid data regarding actually not only people drinking coffee, but some people focus on people, the young kids, or not young kids, the pregnancy woman, while they're drinking coffee, does it cause anything? So far, people don't find anything that bad caused by the drinking coffee. Rather, the only solid evidence showing that pregnant women drinking coffee actually reduce the baby's risk of having type 1 diabetes for some reason. No one give it any mechanism at all. And but by the way, I'm just telling the truth, the knowledge I know. I'm not really encouraging you to drink coffee if you're pregnant or anything. So this is about, yeah, let me see, 3 example of the neurotransmitters. And then let's talk about really something really big, glutamate and GABA. And why this two is important. These are the 2 most abundant neurotransmitter in the central nervous system. And their function is to activate the neurons, and this to inhibit the neurons. So basically, whenever, the neurotransmission is basically on and off. So We have a very sophisticated thought in our mind, but that's because the neural network in our mind. And if you only look into two neurons, one neuron to another, there's only two options: activated or inhibited. And glutamate is activation neurotransmitter, and the GABA is the inhibitor neurotransmitter. And in the glutamate, we need to do some further say introduction. AM glutamate can bind to two type of receptors in the downstream neurons, AMPA and NMDA. Okay? So these are two types of receptors can receive a glutamate, and this is acute and acute activation by the glutamate, and also really only need a low memory potential. A low memory potential low memory potential? Yes, it's only activated by low memory potential. And this is only when you have a repeated or long-term activation by the glutamate. And you need a high potential to activate NMDA. And then what does AMPA do? AMPA just merely activate the neuron. But what NMDA do? NMDA is go to gene expression to let the cell to express more AMPA. And this has a very significant effect on the long-term memory. So in my notes, I introduced later in the later states, but let me do this for now. So in the notes, I talk about the difference between short-term memory and the learning memory and the long-term memory. In all of those, the major difference here is the most important here is the long-term memory. How we have the long-term memory, and the basically is what is memory at all. Let's say you have this very simple model here. You only have 9 neurons here. What is a memory? For example, the two people look into a round thing. Okay, into a round thing. One people may go to think about a sport, say basketball, football, soccer, whatever, and the other people think about space, and another people think about food, donuts, bread. Why it happened? Why this kind of this going one feed? It go to different paths. That's because sometimes it go this way. That's a memory. Or for the other person, they go this way. And for the other person, they go that way. And what determines which way to go? So the key thing here is when we trying to change our memory, we remember things. There's no new neurons to be generated, or no killing any neurons. So basically what we do is we rewire the connection between neurons. Let's call it synapse. Class T, C, D. And at the beginning, at the early study, people believe that the synapse plasticity, that's basically you are trying to produce new synapse and remove new synapse. And if one, say, for example, this guy watch a lot of sports, and this pathway being reinforced again, and the connection to the other neurons is being lost. However, the new research showing that it's not the case. So synapses are still there. However, what's really changed is that the AMPA. How many AMPA are there? So if this guy watch a lot of sport there, then more AMPA is formed in this way. And think about it, you are dumping some water through the heel, and this path is wider, and then it's very easy to go this path, and very easily to go to the sport path. And for people say they really like to eat, for example, me in my class, I like to talk about food all the times. So I think about food all the time, and then when I think about this food all the time, this my AMPA, because repeated activation of this pathway, NMDA are activated, and then NMDA produce more AMPA in this pathway, and then this pathway become much easier for the signals, for the signal transmission to go through this pathway, and eventually this pathway it became easier. It form a new pattern of thinking, or even new memories go into this way. So in this cases, then you have a new long-term memory. So the current hypothesis based on this is called potentiation. The fundamental mechanism of potentiation is that when you repeatedly keep activating one pathway in the brain, the NMDA upregulated, and then the NMDA is activated, and then it make more AMPA on the surface of the neurons. And this neuron is much easier to be activated by the glutamate. So yeah, that's it. And fundamentally, GABA is inhibitory. Glutamate is say excitatory, and then the long-term memory is mediated by the making the neurons more sensitive to glutamate. And next slide is, oh, I'm sorry. Okay, let's talk about something called summation. What is summation? We just learned I draw a neuron here, this cell body, and this dendrites, axon. So most of the synapse happen here in the dendrites, and so we just say now this is a chemical. This is a chemical. Let's see. It is a chemical synapse. Now I use the red dots indicating the glutamate. So the glutamate activate this dendrite synapse. So the question is, if this dendrite is being activated, is the whole neuron being activated? The answer is no. Because there are, firstly, what happened here, that the activation of this dendrite is only got some of those a little bit positive ions get into the cells, and then this ions, sodiums and anything else, is moving forward. I use the red into the sodium ion also, and those ions get in to here, and at the same times, and these ions are not enough. So here, this is the HELOC here. And at this moment, if this ion got in here, increase a little bit, but not to the threshold yet. How you can get to the threshold? How about here? you have another activation, and then more 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 sodium get in to increase the threshold a little bit, almost there. However, something happened on the other side. Now I'm using a blue one. Now here is this one is a negative, and then the negative coming here, it getting into the chloride here, chloride getting in, and at the same time, you lose some potassium. The green one is potassium, the blue one is chloride, and everything coming to here. So this ion passing, this is excitatory, and this is called excitatory synaptic post EPSP. Excitatory excitatory post synaptic potential, and this we call it inhibitory post synaptic potential. And one neuron has hundreds of dendrites, and at the same, it's not unlikely that at the same moment every single dendrite is being activated. However, many of those being activated, and they keep sending this potential, either excitatory or inhibitory post-synaptic potential right way to the helix. Overall, the helix is showing this a little bit higher, and going down a little bit, and going higher, and going higher. Eventually, only that it reach the threshold, you get this axial potential. And then the process that you sum all of those EPSPs and IPSP together in the helix, this is called summation. So any neurons, the activation of the neurons, it only happen if the summation result in the member potential the higher than the threshold potential in the region of the helix or the trigger zones. And then if you reach a member potential higher than the threshold in the trigger zone, you generate axial potential. Once axial potential is generated at the trigger zone, it go all the way here. There's no going back anymore. It automatically going forward till go to the end of the axon terminal to activate another say synapse action. And then I think this may be the last topic of the whole this last view of the never mind. Last thing about this topic. So this remains a big issue. Say, for example, you are listening to me. Probably you are looking into the screen also. But how does this visions and sounds really concentrate together in your mind to be a thought, to reflect as a vision and the sound in your mind? And this whole thing you call neural integration. Neural integration? Yeah. Neural integration or neural coding. Because try to think about it. Everything you see, you saw, you eat, you smell, this is basically the synapse, the electron neuron signals in your mind. How you code this everything into your thought, in that you can feel, you can appreciate. The short answer is we don't know. We don't really know how these neural signals are coded in our mind to reflect as our sensation of the world. And that's very simple. That's only our knowledge regarding this part is, I don't think it's very clear. Many people working on it. And in this class, I only introduce two of those kind of simplest model of the neural coding. One is the rate coding. And for example, don't do it too hard, but you can just try to pinch your own hand You can soft and make it harder and harder and harder, become more and more painful. So anything we have, most of our sensations has a degree, pain, smell, vision, light, all have a grade, all have a level, but how this level is determined. So to our mind, this is one pulse. This is just one signal. The one signal itself has no amplitude. There's no harder or softer. You only have this. Like this one. This is axial potential. You have one axial potential. This is just axial potential. One axial potential cannot be harder or softer. It's just 0 or 1. And then how our body can feel a different pain. Look at this one and look at this one. What's the difference between the upper one and the lower one? The upper one has a lower frequency, and the lower one have a higher frequency, and this is more painful. So our body sense the degree or the level or intensity of the stimulus by the frequency of the stimulus input, but not each single input. There's more input fueling a stronger stimulus. Another thing called population coding. Population coding. So in this way, this is done in cat. It is the researchers like the cat to watch this thing, and this thing, all different directions, and they find in the cat, if they map the brain, this activate this group of neurons, this activate this group of neurons, and this activate this group of neurons. So a different direction of the shapes activate different group of neurons, and they say they all activate some neurons, but where the neurons are activated, they tell me a different story. So this is called population coding. And I think that's all for this topic. Thank you for listening, and see you in the class. Thank you. Bye.